How does adaptive immunity use antibody-antigen binding? Imagine your immune system as a highly skilled detective agency, always on the lookout for clues that reveal dangerous invaders. One of its most precise tools is antibody-antigen binding, a process that helps identify and fight off specific threats. When a pathogen like a virus or bacteria enters your body, it carries unique markers called antigens on its surface. These antigens act like fingerprints, helping the immune system recognize what is foreign. Specialized immune cells, called B cells, patrol your body and have receptors that match specific antigens. When a B cell encounters an antigen that fits its receptor perfectly, it becomes activated. This activation triggers the B cell to multiply and turn into plasma cells, which produce large amounts of antibodies. These antibodies are proteins designed to recognize and bind tightly to the same antigens that activated the B cell. Once an antibody binds to an antigen, it can do several important jobs. First, it can neutralize the threat by blocking the pathogen or toxin from attaching to your body's cells. Think of it as covering the villain's face so they can't cause harm. Second, the antibodies act as flags, marking the invaders for destruction. Cells like macrophages and neutrophils recognize these flags and engulf the pathogens, clearing them from your system. Third, antibody-antigen complexes can activate a series of proteins called the complement system. This cascade can punch holes in the pathogen's membrane, causing it to burst and die. The immune system makes these antibodies even better over time through processes called affinity maturation and class switching. Affinity maturation involves genetic changes that make the antibodies bind more tightly to the antigen. Class switching allows B cells to produce different types of antibodies, like IgG or IgA, which are better suited for different parts of your body or types of threats. Each B cell produces antibodies with unique variable regions, thanks to a process called genetic recombination. This diversity allows your immune system to recognize an almost limitless variety of antigens, making it highly adaptable. When the same pathogen tries to invade again, memory B cells remember the previous encounter and produce antibodies faster and more effectively. In short, antibody-antigen binding is at the heart of adaptive immunity. It helps your body identify specific threats, neutralize them, recruit other immune cells, and remember past infections. This targeted approach makes your immune defenses both precise and capable of learning from experience, keeping you safer over time.